All right, so what determines how all of this stuff is put together and made? I would say nucleic acids are probably, sort of like the king of the hill as far as like molecules go, right, for the body. All the protein amino acid sequences, which determine what proteins do, is coded for with nucleic acids. Okay, so this is a really big one. You probably heard, if this is your first bio class in college, DNA is really, important to understand in order to understand how the rest of any living thing works. Okay. So keep in mind, DNA is, you may have seen it before. It's kind of like this, sort of double spiral, which we call a double helix, right? It kind of goes like this. And it's got these little rungs like a twisted ladder. Okay. DNA, put a couple more in here too. So, is the molecule that basically gives the instructions to a cell on how to do things, right? So, in other words, why does your hair follicles make hair? Because your cells know how to make keratin. Why does your red bone marrow make hemoglobin in your blood? Because they have a gene for it, right? So, keep in mind that that's important. So, there are two types of nucleic acids, right? So just like we have carbohydrates and we have proteins and we have fats, this is the last group, nucleic acids, and they come in two different types. Deoxyribonucleic acid, you'll never have to say that or have to spell it. I would just call it DNA at that point. And then ribonucleic acid, as you can tell, an oxygen has been added here. So that is, and with a couple other differences, that makes RNA. Okay, so DNA provides directions for its own replication and serves as a template so it's replicated with each cell division. Now, what this means here, and we'll get into this when we do cells, which is a future chapter. Every cell in your body needs to have DNA in it, right? That means every time you grow or heal from an injury, you have to make new cells with a new copy of DNA, right? So DNA must copy itself from the first cell to be put into the two new cells that come from it. I think probably everybody in here knows at this point, you have one parent cell, they eventually divide, and you have two what they call daughter cells, right? So DNA has to be put in every one of those, which is why it has to copy. Okay. RNA is made from DNA. So imagine DNA is like, an instruction manual for the entire organism, right? So, it's ridiculously complicated and very long, right? RNA just takes the directions from one of those pieces of information, how to make one protein that makes up your body. I don't know, we'll say keratin, like we said before, and just sort of like takes that information and brings it out to the cell so that the cell can do something with it. Okay, so mRNA is kind of like a tiny little part of the instructions from the whole manual, which would be DNA. Okay, so that mRNA comes out into the cell, and then that eventually allows the cell to translate it. Okay, translation means the cell can use the mRNA, which was made from the DNA code, to know how to put together proteins. Okay, this is called the central dogma of biology. And guys, I gotta say, it's probably the most important thing that you could remember in any biology class. Okay, so what we're saying here is that DNA, what everybody has, will eventually be transcribed, as you can see right here, into RNA. That will eventually be translated into a functional, actual, does something in the body protein. Okay, so this is really important. If you make a mistake here, right, in the DNA, that mistake is going to keep bouncing down the line, and that means you're going to make faulty proteins. We talked about in the last, discussion here that when people have sickle cell anemia, they make the wrong protein in their hemoglobin. Well, the reason that they did that is because they had something wrong with their DNA. They have a genetic mutation that leads to that faulty protein being produced. Okay, so we'll talk about this more in the future, but I just wanted to make sure I didn't blow by these major bullet points here and explain it a little bit better. Okay, so the structure of nucleic acids, uh, just like the other polymers and that we've talked about before, basically they consist of monomers. Now these monomers consist of themselves little subgroups. One is a phosphate, okay? One is a sugar, which can either be ribose or deoxyribose, and one is a nitrogenous base, okay? Meaning they're based on nitrogen, okay? So this is a ring structure here, but you can see that there are some bases that actually have double rings. Okay, and we'll be talking about those as we go. Oh, and actually, before I move on to the next slide, what we should talk about is this monomer. So even though it's basically made of three distinct parts, this is still considered a monomer of a nucleic acid, in this case of DNA. And we call this a nucleotide. When you string nucleotides together, just like you string amino acids together to make a protein or sugars to make a starch, this would be a monomer of the polymer, which we would call nucleic acid. Okay, so what I want you to pay attention to, and what I want you to, understand is really the most important of most important part of nucleic acids. It's not the phosphate, right? The phosphate kind of is like that little circle, right? It's not the sugar, right? So this would have been deoxyribose, and then you have another phosphate and then another sugar. What the most important part of this is the nitrogenous base. It's that one part that can vary in DNA. Everything else in DNA is going to be the same except for the bases, okay? And that the bases are based on nitrogen. As you can see in these pictures here, you see a little nitrogen in each one of the molecules, right? So um, these bases are the ones that vary, right? So if they vary, that means obviously you can have different sections of DNA because of the sequence of these different bases, right? So in other words, you could have for a length of DNA, you could have like A, T, C, C, G, right? What that would mean would be you'd have adenine, thymine, two cytosines, and a guanine, right? They're all running in that direction, right? And these sequence of bases is what determines what protein is going to be built from that sequence. Okay, so we'll get into this much more deeply as we go through the rest of the unit, especially when we get into cells. But you should know that because these can vary, because you can have cytosine, thymine, adenine, and guanine in DNA, as you can see right here, right? You can have a double, a single, 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 right? That means that that variability can build different proteins. And if I'm not explaining that well, if you don't understand, obviously please come into the sessions, and we will definitely talk about it. And hopefully I'll be able to, give you a little bit more insight. Okay, so I wouldn't worry about knowing the difference between pyrimidines and purines. Unless you're going to be a genetic engineer or something like that, then you'll have to worry about it. I think it's a little bit too much detail for us. Okay. But what I would like you to know is that in DNA, right, these sugars here, right, is deoxyribose. That's why I drew the D. In RNA, the sugar actually has one more oxygen to it, and that's called ribose. Remember, most sugar names, they end in ose, right? So you can see them right here. In DNA, you're going to have this one, deoxyribose, and in RNA, you're going to have that one. Okay, so nucleotide polymers, right? So, in other words, these monomers of DNA are linked together, building what we call a polynucleotide, okay? Or you just call it a nucleic acid, all right? Um, these sequences of bases along DNA and mRNA is unique for each gene. I kind of really don't, I'm not really worried that if you know this one or this one, but uh, what you need to know is, since the sugar and the phosphate are the same, that doesn't really matter. Anywhere on a DNA strand is going to have the same sugar phosphate, backbone. What's going to happen though, in the sequence of bases is really what's the most important, as I mentioned last time in the last slide. Okay, so what's the difference between DNA? Well, we already know that DNA has deoxyribose and RNA has ribose. Okay, so those are the two differences. Similarities, though, is that they're both polymers, right? I mean, obviously they have nucleotides that are all strung together, just like any other polymer we've talked about so far. And they're all built from four nucleotide monomers. Now, this is going to make a lot more sense when we talk about the differences, okay? And as we go. I wouldn't worry about this one because we're just not at that level, especially at distance where we're ever going to have to work with it. And I wouldn't really worry about that one either. I would more rather you guys worry about, the differences between DNA and RNA. DNA, the sugar is deoxyribose, and in ribose or in RNA, it's ribose rather. DNA is double stranded. So DNA is going to look like this, right? As we said before, where there's two strands. RNA is going to be a simple line, okay? DNA contains adenine, thymine, guanine, and cytosine. Okay, so all of these bases are in DNA. Okay, in RNA, you have adenine, and instead of thymine, you have uracil. So that's different. Okay, so these are the two different ones. But then guanine and cytosine are the same. Okay, so in DNA, it's A, T, C, and G. In RNA, it's A, U, G, and C. Okay, so I think that's important to know. Definitely want to know the differences between DNA and RNA. If you decide to go on and take Bio 102, you'll do a lot more of this stuff, by the way, looking at sequences of nucleic acids. Okay. Um, because DNA is double-stranded and RNA is single-stranded, RNA is way more unstable than DNA is. In other words, what I'm saying here is that it changes quite frequently in living things. Okay. And hopefully to drive this home, if you think about it, we need to get new flu shots every year. And I know, everybody's thinking one way about vaccines or another, but the reason we have to get flu shots every year is because flu viruses are based on RNA. And because they're based on RNA and RNA is very unstable, they can change from year to year. So sometimes you might get a really mild flu, sometimes you might get a really severe flu, right? And because they change, we are burdened with, having to make new vaccines all the time to make sure that, we're being immunized against, the actual strain that's spreading around. Okay, so RNA viruses like the flu are extremely unstable, and that means they can mutate or change really, really quick. DNA-based organisms like us, very, very stable molecule, right? So we'll say stable. They don't change that much, so it takes a really long time for anything based on DNA to actually change and therefore evolve or mutate and evolve. Okay, so this is a little bit of a side here. So the linear sequences of DNA are passed from parents to offspring. I don't think that's any, surprise to anybody. If you think about, why you look like your parents is because you share some of their DNA. Why you look like your brothers and sisters, same thing. You don't look exactly like them because, keep in mind, this is all a mixture of nucleic acids, but you do look a little bit like them because you do share some certain genes, right? In two closely related species, DNA is more similar than in more distant related species. So we can use this to actually classify living things, as we had said back in chapter 1. So we can also see how they may have evolved, and that's using molecular biology to figure out where the direction of evolution may have happened. Okay, the last slide in this whole presentation, this is 51 slides, so you guys are, I think we're all doing really well here, is about the central dogma of molecular biology. Okay, this is what I said before, and I just kind of want to highlight this again. The DNA in the cell, or in the nucleus of the cell here, right? That's the list of the entire instructions of a living thing. Everything on how to make, if this was human, how to make your fingerprints, all the way how to make your brain, your heart tissue, everything is, basically coded for in the DNA. In mRNA, this is just a copy of one little tiny section of your DNA, right? In other words, it copies for one protein. Okay, so not very big. It's just a small piece of information that your body can use to make that protein. So once that little tiny bit of instruction on how to make a particular protein comes out of the nucleus and into the rest of the cell, don't worry, we'll talk all about all this. Your cell uses a little machine inside, a little organelle called a ribosome, to make a new polypeptide. In other words, a new protein. See these little amino acids here? Your ribosomes, these things, are using the code on RNA to put these in the right order. Okay, so that means your protein will be made correctly as long as your DNA is correct, right? Because your mRNA is made from the code in your DNA, right? So this is how every living thing on the planet, viruses all the way up to human beings work. And I think that's important to know. But don't worry, we'll be hitting this in a lot more detail later. I know this was kind of like a really quick run through of all the large biological molecules in the cell. But again, like I said, if you have any questions, let me know. And you also have a lab dealing with this kind of stuff too. Okay, so hope you have a good one. That's it for this chapter. So we're done. And I will post the next one next week.